Hello everyone. Hi, I'm Bhargavi and welcome to Biodrunk. So, we are going to learn something new about biotechnological approaches here. What is biotechnology? In today's class, we are going to learn. Okay, and in every classes, you're going to get a PDF even to know what is your learning outcome. Okay, and some theory part even. So, welcome to the class, and let's start with our first chapter, that is Introduction to Biotechnology. And if you like the video, do subscribe because the upcoming videos would be in a pattern where you're going to learn each and every aspect of biotechnology one by one with the notes. So, let's start. So let's start with the basics of biotechnology. So when I talk about biotechnology, let's introduce it. What is it? Okay. So biotechnology is nothing but it is a fusion of biology and technology. Okay. So what does this mean? How is this a fusion of biology and technology? So what is biology? Biology means study of biological agents. So here I'm using the biological agents, okay, to produce something that is useful. So to generate a useful product, I can say that. So this is what forms a word or a new sector that's called as biotechnology. So easy. We just learned the definition right now within seconds. Okay. Whenever someone asks you what is biotechnology, you can directly say biotechnology is the use of biological agents to generate a useful product. See, only the two approaches, however you're writing the definition, you need to mention the two approaches. That is, it is utilizing a biological product such as microorganisms or higher organisms, whatever they are living or dead. Okay, or their components such as enzymes or nucleic acids, anything. Okay, in such a way that it generates a useful product, and useful product is to enhance the human welfare. Okay, so this is such an easy definition of biotechnology. Let's now talk about its different uses, biotechnological uses during different phases of human evolution. So first one was old biotechnology. When I'm talking about human evolution, don't take it as the human evolved from monkeys to the chimps, some thought. Okay, I'm not talking about that. I'm talking about the way the humans took biotechnology. Okay, so whenever we talk about old biotechnology, you might have heard things like wine, okay, bread. These are all the things which you use in your daily life. Okay, so wine, bread, and let's talk about something more like curd. Curd is such an easy example. You can see it in everyday life. Okay, curd, yeast. Yeast is also found here. You most of them make chole bhature with this. Okay, yeast. So these all are things are been made since 5000 BC. So 5000 BC, since then the people are using this biotechnology and no one knows it. Why do we call it as old biotechnology? Because they are not using microorganisms. It's the natural process. Here the microorganisms are growing naturally. They are not being used. Okay. So I can say that it's the natural capabilities of the microorganisms. Okay. It's the natural capability of the microorganisms. So we considered it as an old biotechnology. Now, see, old biotechnology, we just did it. That's also called as traditional biotechnology. Now, the in between the modern biotechnology and traditional biotechnology, there was also a phase which started the use of microorganisms for commercial production. So what are we doing here? We are using microorganisms for commercial production. Okay, so when I say commercial production means huge quantity production for something. Okay. So, for example, let me give you one example. First World War. Okay, so during the First World War, it led to the synthesis or production of many new things. Okay, so for example, let's talk about the first one where Germans had to produce glycerol. Okay, so why are they producing glycerol here? Because they had to produce glycerol because their British Navy has disrupted their vegetable oil production. So that's the reason they had to do that. Okay. And remember, glycerol is used to produce or used for manufacturing explosives. Okay. So first they used vegetable oil as the British Navy didn't allow them, so they started with glycerol. In the same way, the Germans also restored few things. They stopped few things that for British. What did British do here? See, the British Navy had to use the production of acetone and butanol. Again, this is for the productions are for the supply of these chemicals to manufacture something like explosives. Okay, so here also this happened. Now, this was during the World War. Now, after the World War, what happened is the citric orchards in the countries were ruined. Okay, so now they need citric acid. So they extracted citric acid. Okay, they extracted citric acid from a microorganism that is called as Aspergillus. So here I'm not writing in Latin. Okay, so it's Aspergillus niger. Remember, it was so easy. So you can also remember the production of penicillin. It's an antibiotic. Okay, and it was also produced from penicillin notatum. So this was what happened here. Since then, man had his quest to improve the natural capabilities of microorganisms, and he wanted to produce something of novel and discover the capabilities of the microorganism. And this is where took a leap to recombinant DNA technology, okay, or gene cloning. So now it led to the modern biotechnology. So it's just a phase between an old biotechnology and modern biotechnology, where we started the use of RDT, recombinant DNA technology, gene cloning, genetic study, okay, functional and structural studies of what? Functional structural studies of nucleic acids, proteins, okay, and lot. So this is where we started a new era where we are creating something new. Okay, we are using microorganisms, we are converting them into some useful product. Here also it's the same thing is happening. Okay, so always remember, biotechnology is a multidisciplinary in nature and it has several disciplines as we discussed before. So whenever we talk about how the processes are being done, okay, so this biotechnological process is majorly divided into 5 steps or 5 operations. The first one is I need a strain. Obviously, if I get a strain, then only I can work on it. So I need a strain. Once I get the strain, I need to improve its quality. So I'm using strain, choice, and improvement. There I need to improve the strain. That means isolated products. So here I'm using microbiology, cell biology, botany, zoology, genetics, so many things. Now I got a strain, I need to get its mass culture. So I need to get it in huge amount. So that is where I'm using mass culture. And then after doing that, I have to optimize its responses. Okay, so then I'll be going for the optimization of cell responses. Now, then I'll do some other processes where I'm going to check how it is working, what is its way of working, how which gene is been producing a particular enzyme, what is happening here. And then once I get it, I'm gonna purify the product and take it. So downstream processing. So these all are the 5 major steps that occur during the production of any new product. Okay, so this is such a easy thing. We always think, okay, they might be made up of a lot of things. Yeah, obviously it has many steps inside it, but yep, we are going to get the best out of it. So what's the scope of biotechnology now? Till now, you have understood what is biotechnology, when it started, how it evolved, okay, and what are the major steps. But now let's know about the scope and importance of biotechnology. See, it has been emerged into an very great big area where we are creating something very new for our human welfare. Okay, it's like producing something important. So just a recent example, COVID, everyone has heard of. So what we did, diagnosis using RT-PCR, production of vaccine, yeah, study of the virus. Yes. So we did so many things just now. These all included microorganisms. This is the very basic example and the recent example I'm giving where the biotechnology is being used. So this happened because we are using it to create something new. So here I am utilizing a product, biological agent. I'm researching on it, I'm studying on it, I'm creating a product which is of very use. Yes, so it's very easy. See, and again, if I talk about what are the different fields, so you can count and there would be many. For example, let's talk about something which we haven't talked yet, marine biotechnology. Let me write it as BT, it would be better. So marine biotechnology, what's happening in this marine biotechnology? We are getting something out of the marine life and we are finding it. Medical biotechnology, just now we discussed about it. Okay, medical biotechnology, industrial biotechnology, that is huge scale production of something. Okay, and then you have enzyme technology. So what is this enzyme technology? Here we are producing something of, for example, I have to produce a fructose. Okay, fructose corn syrup. So I'll be using an enzyme called glucose isomerase. So this is how I'm using enzyme here. So enzyme biotechnology. And then protein. Protein or enzyme engineering, enzyme biotechnology could be nearly same, but as all proteins are not enzymes, but all enzymes are proteins, so it differs. So I'm writing it here. Protein biotechnology. So this was all about biotechnology. Hope you understood all the concepts given here and you liked the video. So for the upcoming videos, do like, share, and subscribe. And this video is also available in other languages like Hindi and Telgu. So you can also get the link from the description box below. So let's meet for the next video. Bye.